Answering MCAT questions in under 60 seconds. A scientist investigates the effects of a viral infection on host gene expression. The virus is found to upregulate a cellular splicing factor involved in exon skipping during pre-mRNA processing. Which of the following outcomes is most likely to occur in infected cells as a direct result of this activity? Okay, let's look at the answer choices. So A says increased inclusion of all exons in mature mRNA transcripts. Well, this doesn't sound right? Because they're saying that you have upregulation of a splicing factor that causes exon skipping, okay? And we just have to recognize that exon skipping is something where an exon that is normally included in the mature mRNA, or the mature mRNA, right, is actually not included. So this is the process of alternative splicing, where basically the same pre-mRNA can yield multiple different types of mRNA encoding different types of protein due to different combinations of exons, okay? And so we wouldn't have increased inclusion of all exons. In fact, we would have increased exclusion of certain exons, right? So this is wrong. B, decreased variety of protein isoforms produced from individual genes. Right, well, this one actually is also wrong because we would actually expect an increased variety, not a decreased variety. Okay, because if you're having alternative splicing, you're promoting various new mRNAs to be produced from that same gene. Okay, and so you should have more isoforms of the protein. And then C, increased production of alternative mRNA transcripts from a single gene. And that sounds correct, right? Because that's exactly what they're doing when you have alternative splicing, is you variably splice the same transcript and create different combinations. And of course, as a result, you'll have different types of mRNA, all encoding for unique proteins, right? So C sounds like the right answer. Okay, but then D says decreased efficiency of mRNA export from the nucleus to the cytoplasm. That doesn't have anything to do with this alternative splicing process, right? The efficiency of mRNA export doesn't change. It's just that you'll have a bunch of different mRNAs being exported, right? So C here is going to be the best answer.